So what I would like to share with you is really of our vision to use the immune system to fight cancer. We are clearly making substantial progress. The immune system has been fantastic in defending our bodies against infections. And in fact, it is due to vaccination activating an appropriate immune response that some of the pathogens which have been a major health burden have been completely eliminated, such as smallpox, for example. So what I would like to do today, I would like to give you a short introduction to the cancer problem. And then I would like to give you a short overview of the key components of the immune system. And then I'll review with you the vaccination approaches that have been very successful for some of the cancers. And then we'll talk in a little bit more detail about the cell and gene therapy approaches that we have been developing here and that are also being developed worldwide. And then we'll conclude, and then hopefully we'll have a good discussion. And if you have any questions as we go along, then please raise your hand. It's absolutely fine to interrupt. I think it's better to keep the understanding rather than sitting back and waiting to the end. So let's start then with some of the introductory remarks to cancer. It's clearly a big problem. It's a lifetime risk of developing a cancer is 1 in 3. So typically once we approach age 60 and above, then statistically it's 33% of the population developing some type of a cancer. In addition, of course, there are also cancers that set on much earlier in life. There are some of the childhood cancers and some of the cancers that occur in adult life. So cancer then, it's clearly a worldwide problem. So every year, approximately 10 million individuals are diagnosed with some kind of a cancer. And I think it's quite interesting to notice that it's not a disease of the underdeveloped countries, or it's not a particular disease of the developed country. Cancer really spreads across the world. And if we look here, it's approximately 5 million new diagnosed individuals in the developed world, and this very similar number in the developing world. So it's clearly then a huge challenge to find new and additional treatments to help with the management of this problem, and also to develop preventative approaches that might prevent the occurrence of the cancer in the first place. So let's now have a little think about what cancer actually is and how it works. So if we look here at this individual, then I think it's useful to realize that each of us, we are not just one individual. We are a collection of cells, and we are a collection of approximately 10 to the 13 cells. So this is a 1 with 13 zeros, so it's a number that is far bigger than most numbers that we are used to. It's a number that's bigger than the world population. So this is what we are. And how does cancer work? Well, cancer originates from one individual cell. And although the risk of developing a cancer is relatively low for an individual cell, but since we've got 10 to the 13 of those cells, then of course the risk becomes much bigger, that there may well be one of those cells that undergoes what we call the malignant transformation and doesn't obey to the normal rules that govern the interaction between cells in a body, and disobeys and grows in an uncontrolled fashion. And that's of course what cancers do. So they multiply, they divide, they grow in an uncontrolled fashion that is no longer regulated by the normal interaction between cells. So then typically a cancer starts with a local growth, and once it reaches a critical mass, then those cancer cells, they start invading the normal tissue. And it's typically at this stage where we start noticing them clinically. And then there's the issue of metastases. And some of the cancer cells, quite often, they acquire the ability to spread within the body. And then they seed as metastatic lesions somewhere else in the body, here depicted probably in the lung. And then, of course, we can also have a metastatic lesion somewhere else in the body. And this is really sort of the biology of cancer and the cancer progression. Now, what are sort of the causes of cancer? What do we understand about it? We know that cancer sort of originates from single cells. And we know that the human genome, each cell, each of those 10 to the 13 cells, has one whole human genome in the nucleus of the cell. And the human genome contains approximately 40,000 genes. So those 40,000 genes, they really define us as the human species. So all those genes are present in all cells in the body. However, in any individual cell, in a lung cell, for example, or in a brain cell, a subset of those genes is active. So it's approximately 10,000 genes are active in an individual cell. And those 10,000 genes that are active in a liver cell are different from the 10,000 genes that are active in a brain cell. And it's exactly those 10,000 active genes that define whether a cell is going to be a liver cell or whether it's going to be a brain cell. So the gene activity defines the cell function. Those genes, they also regulate the growth of the cells, the frequency of a cell division. A cell division results in cell multiplication. And it's really this regulation that goes wrong in cancer cells. So we've got an abnormal gene activity. Some genes are active at much higher level than in normal cells, or they are active at reduced levels. And some genes are mutated in the cancer cells. So it's this dysregulated activity of the genes that results then in an increased cell division or in an increased growth of those cancer cells, which results then essentially in the tumor growth and in the clinical problems. So this is essentially some of the baseline biology of cancer. So we do understand now that cancer, in essence, it's a genetic disease. The genes don't work properly, they are dysregulated, and sometimes it's also genes that come in by viruses that contribute to the dysregulation in cancers. So what about the immune system now? Let's have a look at the basic components of the immune system. So the tasks of the immune system are really to protect the integrity of our body. And in that sense, it's strictly defensive. It reacts when there is a foreign invader. And typically, those are viruses that are trying to get into our body. Those are bacteria or other kinds of pathogens. And when those invaders enter the body, then the immune system is typically very effective in an efficient attack and in the elimination of those invaders. And at the same time, it's important for the immune system to be tolerant to our normal body, to our normal self, to the cells, to all those normal cells that are present in our body. And of course, we understand that sometimes this tolerance regulation is defective. And if tolerance is defective, then individuals can develop sort of autoimmunity. So tolerance and not attacking a normal body is one important aspect of the immune system. And it's one aspect that I'll come back to, which can also impair the immune response against cancer cells. Okay, so now if we look at the cell types that help the immune defense of our body, we've got major cell types are for broad range defense are the granulocytes and macrophages. And then for a smart attack, we've got lymphocyte populations. We've got B lymphocytes and T lymphocytes, or B cells and T cells. And there we also have got those so-called killer T cells. And those cells are very effective in killing off cells that harbor a virus, for example. And those are also the cells that we would like to recruit into the cancer treatment, to direct those killer T cells to selectively recognize and attack and eliminate the cancer cells. So here we've got then the sort of the development and the function of the immune system. The immune cells, they all develop from a stem cell, and this stem cell lives in the bone marrow. This stem cell then grows and multiplies and develops into those cells that give rise, that develops then into the macrophages and neutrophils. And those type of cells are called innate immune cells. Now the feature of those cells is that they can recognize quite a large range of viruses and bacteria. And when one of those viruses or bacteria enters the body, then those cells get activated and make an immune response. And that is different from those cells over here, those lymphocytes, those T cells and B cells. Those cells, they mount what we call this smart attack. So only some of those T cells will recognize one virus very specifically. But once they have seen this virus, those cells will remember that this virus has been around, and it creates what we call this immune memory. When the virus comes around again, then it's attacked and it's eliminated much more rapidly and much more effectively. So let's have a look now at sort of the immune systems in the human body. We've got those stem cells. They live in the bone marrow. And then some of the immune cells go into the thymic glands that sits here just above the heart. That's where they are trained to become those T cells, including those killer cells. The other immune cells, they actually develop in the bone marrow and leave then the bone marrow as monocytes and macrophages and neutrophils, or they leave the bone marrow as B cells. So this is now sort of a picture of a lymphocyte. You can see here the center of the cell, that's the nucleus. This is the space that contains all the genetic information. All those 40,000 genes are stored in the nucleus in the form of chromosomes. And then we see the cytosol here, and then the surface of the cells. And on the surface, the immune cells, they typically have structures. They've got molecules that allow the immune cells to recognize bacteria or viruses. And they have structures that potentially allows them to recognize cancer cells. And it is really those structures that we need to identify and exploit, that the immune cell can selectively recognize the cancer cell. So here we've got now a little video clip of an immune cell in action. And I'm just going to play that for you. Here we've got a neutrophil. And here you can see there is those little dots here. They are bacteria that have been put into the culture containing this neutrophil. And you will see how this neutrophil chases the bacteria and tries to eliminate it by eating it up. So here you can see the bacteria moves around. It keeps escaping. The neutrophil keeps going after it. Turns around again, and it moves up, tries hard, and now there we go. So that's essentially what our immune cells can do very effectively, to spot that there are foreign invaders there and to attack them with persistence. Now let's briefly have a look then of what sort of the B cells are capable of doing. So this is one component of our smart immune system. So the B cells are the immune cells that produce those type of molecules, and those molecules are called the antibodies. And those antibodies, they have a structure that allows them to selectively recognize structures on a bacteria or virus, or to selectively recognize a structure on a tumor cell. And it's useful to think of those structures as keys that fit into one specific lock. So if those immunoglobulins go around and they can't find the appropriate structure that matches this key, then they won't act and they won't be able to provide protection. And then vaccines, they exploit the ability of the B cells to produce those protective antibody molecules. So we get vaccinated with dead viruses, or some vaccines are also based on live virus vaccines, or some vaccines are based on cancer cell vaccines. And the rationale behind it is to now selectively activate a particular B cell that has the correct structure, the correct antibody, that can efficiently recognize the component in the vaccine. And in this case, you can see it's this type of structure that can bind to the vaccine component. Those cells get activated and they produce a large amount of those antibodies that can bind to the vaccine component. And those antibodies then can also bind to viruses if they enter the body. And it is those antibodies then that can provide protection against the virus infection. And this is just a little picture showing that vaccines have also been used successfully now in the prevention of cancer. And it's particularly effective, of course, for those type of cancers that are linked to a virus infection or to a bacterial infection. And here is a list. Here are the type of viruses that can cause cancer in humans. It's the hepatitis viruses, hepatitis B and C, that in chronic infected individuals, they can cause liver cancer, which is also called hepatocellular carcinoma. It's the human papilloma viruses, particularly type 16 and 18, but there are also other types. So this type of virus causes cervical cancer. And then you can see there are another few viruses, and there are also some other bacteria and parasites that can cause cancer in people. And it's those type of cancers that have been successfully attacked with vaccines. So we've got an example where the vaccine against the hepatitis B virus has been introduced and has drastically reduced the infection rate by this virus, and also has drastically reduced the frequency of liver cancer, in particular in Asia, where liver cancer caused by chronic hepatitis virus infection has been a huge problem, and the hepatitis vaccine has made a substantial contribution in reducing this disease burden. And then you probably all know that now in the UK and in many other countries, we've also introduced a vaccine against the human papilloma virus. And it's our schoolgirls who are being vaccinated, and it will protect them against the infection with this particular virus that can cause cervical cancer in women. So it's very likely that this vaccine will be effective and will reduce cervical cancer incidence in the future. Now the vaccines, as I told you, they are based on the antibodies. And here we've got another picture of an antibody molecule, and it's a golden antibody, and it just indicates that antibodies have been very successful in the treatment of cancer. So the antibody molecules, they have been targeted against structures that are selectively or primarily expressed on cancer cells and not on normal cells. And quite often those structures are the same structures that we find on normal cells, but they are present at much higher levels on the cancer cells. And in order then, and here we see, we just see a picture here of those antibody molecules attacking those surface structures that are present on the cancer cell. And as I said, sometimes those structures are actually also present on normal cells. And as a consequence, the patient's own immune system is unable to make those type of antibodies. So how do we get around this then with the antibody type approach? Well, most of the antibodies are actually made in animals. And indeed, it's very frequently mice, and it's also other type of animals. So those animals, they are immunized with a particular structure that is present on the cancer cells. And since it's a human structure, the animal, of course, is not tolerant to this structure. So the animal makes an effective immune response, and then those B cells, they are isolated and cultured then in the laboratory or in the pharmaceutical industry. They are also cultured and then purified to isolate those antibody molecules, which then have the very defined specificity targeting the particular structure that has been used to immunize those animals. And antibodies certainly have revolutionized the treatment of a number of cancers. It's a number of lymphomas, where antibodies such as rituximab have been really very effective. And you can see how industry has also been very fast and very productive in developing of antibodies as a therapeutic option for our patients. You can see some 10 years ago, it was a relatively small market. In 2010, the market for antibody-based treatment, and most of the treatment is actually in cancer patients, was worldwide approximately $25 billion US dollars. So now I would like to go on to the next generations of immune interventions that we are developing. And rather than using those antibody molecules, we would like to use those type of killer cells. The killer cells to direct them to selectively interact with the cancer cell and to develop then the killing activity targeted against the malignancy. So how do those killer T cells work? Okay, they are the type of lymphocytes, and they have on their surface this particular key. So it looks a little bit like an antibody molecule, but it's called a T cell receptor. And this key, again, it fits into one particular lock. It can fit into one particular structure that is present on the cancer cell. And that gives it then a highly specific biological activity. So this receptor molecule, it is required for the specific recognition that this cell can undertake. Each killer cell has a unique recognition structure. And as a consequence, there are many, many different types of recognition molecules present in the immune cells of our body. And this is illustrated here. So if we look now into the type of killer cells that are present in our body, each killer cell has a different key and has the ability to recognize a different type of structure. And how does this system work then? If there are such a huge diversity, how do those cells know when to mount an immune attack? So if now a structure comes along, it can be a virus structure or it can be a structure on a cancer cell that binds to the key on the killer cell, then this killer cell gets activated. And it starts growing and it starts dividing. And now suddenly we've got a large number of killer cells. All of them have the same key. all of them have the ability to target the same structure, which may be a virus structure or it may be a structure on a cancer cell. And this makes this system so potent. A relatively small number of cells can be very effective because they can grow in the body, they can multiply in numbers and adjust their activity based on the magnitude of the problem that is around. Okay, so what makes it then difficult? What is the problem? So what's the problem with our killer cells? Why don't they consistently attack the type of cancer cells that may develop in our body? One of the problems is that the cancer cells, they are probably 99% identical to our normal cells, to the normal body. And we've learned that it's very important for our immune cells to be tolerant and not attack our normal cells. And that is the difficulty. There are only a few genes that are dysregulated in the cancer cell, but apart from that, the genetic profile of our cancer cells is very much the same as the genetic profile of our normal cells. And that makes it difficult then for our killer cells now to mount an effective attack against those cancer cells. So the type of approach then that we've developed to overcome this problem is to recruit killer cells from a healthy individual. And the rationale behind that is that we know that the immune cells in each individual is tolerant to your own body, but your immune cells are not tolerant to organs or to cells of a different individual. And of course we know that because that's partly why transplantation is a problem. When we transplant an organ from one individual to the other, then typically the immune system tries to attack this organ and tries to reject it. So what we have been doing then to use this observation, take the immune cells from the healthy donor and now select those type of killer cells that have the right keys, that have the right specificity to recognize selective markers that are expressed at high levels in the cancer cells of the patient. So essentially we've used this design to overcome the problem of tolerance, to isolate killer cells that can effectively recognize cancer cells that have arisen in a patient and mount that way an effective immune attack. So here is now sort of the overview of sort of the novel type of immune therapy that is now sort of entering clinical trials. So I've just explained to you that we isolate the immune cells from healthy individuals. And what we do is then we isolate now the genetic material. Here we've got the DNA. So this immune cell now has the genetic code that is responsible for this key. So this key is programmed in this genetic code. So we isolate the genetic code and transfer the genes into the immune cells of the patients. And now the immune cells of the patients, they start receiving now this structure. They start receiving now the key that allows them to selectively recognize the structures on the cancer cell. So then those genetically enhanced immune cells of the patients are used now to fight the patient's own malignancy. Now it's been sort of a long road. You can see though we've started developing these concepts of more than 10 years ago. And we had to do quite a lot of experiments sort of in the laboratory. We had to be able to isolate from the healthy individuals the killer cells that have the correct keys, that they can selectively attack the cancer cells. And then we had to isolate the genetic material that is responsible for this key. And then we had to develop the gene transfer technology that allows us to transfer this genetic information into the killer cells of the patients. And now we are there. And some of this, and some of our experiments that we had to do before we were allowed to try this treatment in patients also involved the proof of concept. So we had to take immune cells from the patients, insert our therapeutic genes, and then show that those immune cells can prevent now the growth of human cancer cells in an animal model. So animal experimentation is a key component of research moving into patients in order to get them approval from the UK regulators. There are a number of regulators that need to be informed, that need to look at all the documents that we have produced about our research, and then the regulators will say, yes, your research has met all the criteria, you have all the proof of principle studies that are required, you can now try this type of treatment in patients. And for us it's very exciting because that's exactly where we are now. So we are now at a stage where we've actually opened our first clinical trial using those type of gene-enhanced patient cells. Now, at the same time, this type of concept has also been developed by our collaborators and competitors in the US. And it's very gratifying to see that some of the small-scale clinical trials that have been performed with those genetically enhanced patient immune cells have shown good clinical efficacy. So they've clearly shown that the patients benefit from this type of treatment. And you can see those studies have just been published recently, just last year and the end of last year. Here a study with gene-enhanced patient cells, which were successful in patients with melanoma, in the patients with synovial cell carcinoma. And here we've got a study with gene-modified, gene-enhanced patient cells that have been effective in patients with advanced leukemia. Those were patients with a form of leukemia called chronic lymphocytic leukemia. So it's a very exciting field to be in. And here is where we are with our pipeline of new types of therapies at the Royal Free. It's a joint development between Royal Free and the University College London. So we have opened our gene therapy trial for leukemia. So this is a trial that has been financially supported by the Leukemia and Lymphoma Research and also by the Department of Health. And we are very proud that we've been able to open this trial last month. We've also have funding from the Medical Research Council to open a trial in transplant patients. And we hope to be able to open this trial towards the middle section of 2012. In this case, we again will use a patient cell, enhance their functional activity with a gene transfer that those transplant patients are protected against virus infection, which is very often a problem in the transplant patients. And then we've got in our program, we've got gene therapy approaches for melanoma, gastric cancer, and esophagus cancer. And this is a program that is funded by the European Union. And there we hope to be able to open this trial in 2013. So in this trial, patient T cells will receive the genetic information that allows those patient killer cells to selectively attack melanoma, gastric cancer, and esophagus cancer. And then finally, we also have a research program to develop the relevant keys and the relevant genetic information to enhance patients' killer responses in a cancer, in liver cancer, and in nasopharyngeal carcinoma. So you can see that each type of malignancy probably requires a different type of key. And those keys, that is our research program, which we try to identify the best possible key for each malignancy. And those keys, they are hardwired in the genetic information. So then for each type of malignancy, we use a set of genes that we introduce into the patient immune cells, which then enables those patient cells to selectively recognize now the cancer cells and mount an effective immune attack. And then the beauty about this approach is that those immune cells, they are long-term maintained in the patient. So if the cancer problem, if there are lots of cancer cells, we expect that those immune cells grow, they divide, they multiply in the patient. And then as the tumor load decreases, or if the tumor is eliminated, then those immune cells will decrease in number. And if there would be then a reoccurrence of the disease, then those immune cells can expand again, they can grow again and multiply again. And this is a feature of what we call this memory response of the immune system. It's sort of a very attractive component of this type of cell therapy. If successful, it should be applied once, and it should be able to give the patient long-term protection. So now, the concluding slides, immunotherapy of cancer. I've reviewed with you that immune molecules have been very successful for quite a number of cancers. And those immune molecules are the antibodies. They have revolutionized cancer treatment for a number of cancers. And they have been really a success for the patients, but they also have been a success for the pharmaceutical companies who have engaged with this immune concept and have developed it very successfully as an effective treatment option. So the immune cells, this is a much younger field. It's very much a field maybe where the antibody production was some 10, 15 years ago. So using immune cells now is a new concept to achieve long-term protection. We've got effective protection in the preclinical models. There are some first promising data from small-scale patient trials. The Department of Health and the government anticipates that the value of cell therapy in 2014 will be approximately 3.1 billion. And it's exciting for us at the Royal Free Hospital and UCL to be really at the cutting edge of this novel type of therapy. And really, in order to be able to remain at the cutting edge, that's where our ambition comes in. We are now at the stage to start the fundraising for the Institute of Immunity, Infection and Transplantation. And this is sort of an architect's impression, architect's potential option for the new institute. Here you see the existing Royal Free Hospital in the background. And here you see the design, a possible design for our new institute. So this institute will focus on the development of vaccines for viral complications, and then it will have a very strong focus on the development of cell and gene therapy, including those type of approaches that I've just discussed with you. And one priority is that we would like that our patients have access to cutting-edge therapies that are probably not easily available anywhere else. So I would really like to acknowledge the long-term fundings that we have received from our funding bodies, the Leukemia and Lymphoma Research. It's a charity, and it's, of course, it lives from the charitable donations of our population. So thank you, and thank Leukemia and Lymphoma Research supporting our research over the past 15 years. And really, the research, it requires sort of a research team. And this is sort of our research team, so a number of individuals, postdoc, PhD students, technicians. And my senior colleague is Dr. Emma Morris. She is a hematologist, and her research is also focused on the development of immune therapies. And then it's also important for a productive research group to have good local collaborators. But our collaborators, of course, are also international. We've got a lot of collaborators in Europe, and we've got our collaborators in the US as well. So I would like to stop here, and I hope that we can have a discussion. Thank you.